COVID-19 aid, a global attempt to raise awareness and relief support. I'm Samiha Nujud-Zaman, your today's host for the event. I hope you all are doing great during this pandemic, even though a lot of developed countries have been able to tackle the situation quite in a good way. Yet, lower middle income countries like Bangladesh and India are still facing horrors each and every day. Thus, considering the situation, Committee of Biotechnology has come up with a campaign to raise awareness and mitigate the misconception relating to COVID-19 among people. COB has also launched fundraising program. Other than this seminar, we have competitions such as fiction, non-fiction, write-up relating to COVID-19, idea presentation, etc. for participants to get themselves engaged in it. We also hope to give healthcare supplies to families that have adversely affected by the pandemic, for which we're gathering funds, cooperation with be done on the foundation. Today we have with us Dr. Sara Clohisey. She is a core scientist at the Bailley Group, a postdoc at the Roslin Institute at the University of Edinburgh, which highly emphasizes sepsis in humans. When it comes to her research interests, she is interested in host-pathogen interaction, more specifically the interaction between infections such as influenza A and innate host immune system. Few of her mentionable projects are GenoMICC, genome CRISPR screen in disease susceptibility, and construction of porcine. Side by side, she's a member of Microbiology Society at the Roslin Institute. Before starting the event, I would really like to thank you, the ma'am, for coming here today, and despite of your hectic schedule, it's an absolute honor for us to have you with us. Now giving floor to Dr. Clohisey, I would really like to ask our audience to drop their questions right at the comment section. Dr. Sara Clohisey will try to answer them after her talk. Now, without any further delay, Dr. Clohisey, the floor is all yours.
Thank you very much. I'll just share my screen. Okay, can you confirm that you can see that?
Yeah, we can see it.
Great. Okay, well, thank you very much for that introduction. And thank you very much for the invitation to speak here today. I'm going to be talking about our late 2020, early 2021 paper, Genetic Mechanisms of Critical Illness in COVID-19. So the first thing I want to do is thank the patients and the families who took part in our study, GenomicsC. Without them, we wouldn't have the samples and we wouldn't have the data. They took part at a time of their lives that was exceptionally stressful, and we really thank them for doing that. We also want to thank all the nurses and doctors who collected samples for this study throughout the United Kingdom. It's over 1,000 people who took part in collecting the samples, and hopefully we've managed to name them all in our paper. So to begin, COVID-19 is a respiratory disease that causes systemic inflammation. It's caused by the virus SARS-CoV-2, and when it manifests, the patient will have fever, a loss of smell in many cases, chest pain and shortness of breath, fatigue, so they feel quite tired, and muscle pain. So these are very similar to the symptoms of other viral infections. When COVID-19 progresses, we can also see kidney failure. There are currently 4 main circulating strains of SARS-CoV-2, Alpha, Beta, Gamma, and Delta. And as you may know from the media, the Delta variant is currently overtaking in the UK. It has slightly different symptoms, it seems, to the classic COVID-19 and looks more like a common cold. So it has a runny nose and a headache, but it has proven that it is still quite severe and can still cause death. So regardless of what strain of SARS-CoV-2 you come in contact with, there are various factors that can tip the balance between severe and mild disease. So where in mild disease, you may be at home for a week or two, very, very ill. In severe disease, you may be in hospital and require mechanical ventilation. Factors that can affect your susceptibility to the pathogen, or whether or not you have mild or severe disease, are very varied. They range from things like age. We know that older people are more likely to have adverse outcomes with SARS-CoV-2. Sex, males versus females. frailty if you have any other diseases, such as asthma or diabetes, obesity, which is lifestyle and various genetic changes, and pregnancy, which can cause differences in how your immune system is modulated. All of these things exist on a spectrum, contributing in different ways to your susceptibility to the pathogen. But one thing that lies towards the end of this spectrum is genetic variability. That is that single DNA changes may be able to cause you to go from mild to severe illness. And how do we know this is the case? Well, in 1988, Sorensen et al. Published in the New England Journal of Medicine a very nice study. They looked at risk of death by cause and compared people's risk of death when compared to their biological parent or compared to their adoptive parent. So this was a study of adoptees. When they looked at the relative risk of dying from a natural cause if your biological parent died from a natural cause or your adoptive parent died from a natural cause, they found that there was very little difference, although you were more likely to die from a natural cause if your biological parent did. When they looked at vascular disease, such as heart disease, they found that you were more likely to die from vascular disease overall if either your biological or your adoptive parent died from this, but the biological aspect contributed slightly more. And we know that with vascular disease, there is environmental factors such as diet and exercise associated with how you're raised, and there's genetic factors that can contribute. Surprisingly, at the time, when they looked at death from infection, they found that there was a substantial difference in your relative risk of death from infection if your biological parent also died from infection. So this shows that if your biological parent dies from infection, you have a very increased risk of dying that way. Whereas if your adoptive parent dies from infection, your risk doesn't seem to increase. So we know from this study and from others since that a person's genetics can decide how ill they will become when they're exposed to a pathogen. And as scientists, we want to find out what DNA changes can cause someone to progress from a mild to a severe disease. And how do we do this? Well, we can perform something called a genome-wide association study. The genome-wide association study, or GWAS, looks at individual nucleotides within the DNA sequence and compares them between two groups, the cases and the controls. The cases are the group that you wish to look at, so they're your ill group, or in this case we've called them the purple group. And then your controls are your background population, in this case the blue group. In our mini GWAS here, consisting of only 10 people, 5 cases and 5 controls, we can see that the purple group is more likely to have an A at a specific point in the genome, whereas the controls are more likely to have a G at that specific point in the genome. There are exceptions, for example, this purple person here who has a G, and this means that their purple might be affected by other genes in their genome or by environmental factors. You then make this comparison 3 billion times for each nucleotide that's in the genome. And eventually you have statistics which indicate which genes or which changes in the genome are most likely to cause your purple or your blue phenotype. To perform a good GWAS, you need a well-defined phenotype. In the case of COVID-19, this is severe COVID-19 causing, for example, death or requiring ventilation. You need a well-defined control group. And for a COVID-19 study, the best control group is mild COVID-19. That is, people who have come in contact with the virus and displayed symptoms and tested positive, but did not progress to severe COVID-19 where they required hospitalization or required mechanical ventilation. You also need a large number of individuals, from thousands to tens of thousands of people. In order to achieve this, we set up GenomicsC in 2016, and GenomicsC is an open, collaborative global community of doctors and scientists trying to understand and treat critical illness. So COVID-19 is not our only focus. In fact, we have many focuses outside the pandemic. Genomics stands for Genetics of Mortality in Critical Care. And our long-term aim is to get 100,000 critical illness genomes, that is 100,000 DNA sequences from people who are in intensive care units throughout the world. The focus is not just COVID-19, as I said. The focus is on other diseases as well, such as sepsis, infectious diseases like influenza and pneumonia, and emerging infectious diseases, and this is where COVID-19 comes under our radar. Genomics has been set up in many different countries, but is primarily based in the United Kingdom, and we're setting up slowly throughout different countries in the world. If you want to find out more, you can see our study at genomic.org, and that's genomic with 2 C's. So for this talk, I'll be focusing on results from the United Kingdom, because that's where we got most of our samples and where Genomic was up and running in the beginning of 2020. The first UK case of SARS-CoV-2 or COVID-19 was on the 31st of January 2020, and the cases increased steadily until lockdown was announced on the 23rd of March. Just before lockdown was announced, we were working very hard in order to collect DNA samples from various different sites all over the country. And in April, we were declared an NIHR urgent study, which meant that within hospitals and intensive care units, our study was given a priority to help understand COVID-19 and assist in the COVID-19 response. By mid-August, we had our first DNA sequences. So in this time, we were collecting blood samples and we were sequencing them. 6 weeks after we had our first sequences, we published our first preprint on medRxiv. That was on the 25th of September. And at this time, we made our summary statistics available publicly so that they could be shared with other scientists and added to other analyses to help the COVID-19 response globally. On the 11th of December, we had our first peer-reviewed results published, and you can see these in Nature at this QR code or by Googling Irola's name below. So what did our results look like? Well, first of all, we had our group of patients that we studied, and their demographics were 30% female. They had a mean age of just under 60 years old, which is slightly younger than the expected 65 plus that we expected given the likelihood of older people becoming severely ill with COVID-19. The majority of our samples were of European ancestry, over 75%, and this is because this study was predominantly carried out in the UK, where most people have European ancestry. We also had individuals from South Asian, African, and East Asian ancestries, but as you can see, these made up a minority of our samples. 20% roughly of our participants had significant comorbidities, that is, they had other illnesses before they presented with COVID-19. 3/4 required invasive ventilation, and just over 20% died within 60 days of being recruited to the study. So these last 2 points about invasive ventilation and death do point out that we were really looking at the very severe people here. These were people who were going to have long-term effects and who were very ill with COVID-19. When we had the DNA samples, we performed array genotyping, and we interrogated over just under 750,000 single nucleotide polymorphisms, or SNPs. These are single DNA changes in the genome. And the results I'm going to show to you will be presented as something called a Manhattan plot, because it looks like the skyline in New York, in Manhattan. And on this plot, you'll see the chromosomes on the x-axis, so along the bottom, and along the y-axis at the side, we'll look at the significance. We'll then have a bar across the middle, and anything which reaches over this bar is seen as a significant point in the genome associated with severe COVID-19. Each dot is each individual nucleotide. So we focused on the European samples because they were the most, we had the most numbers of these within our cohort, and this was 1,676 samples. We compared these to 5 times as many controls from UK Biobank, a publicly available group of DNA sequences that were collected within the UK. And we were able to see 7 different points in the genome associated with severe COVID-19 on chromosome 3, 6, 7, chromosome 12, 2 points on chromosome 19, and 1 on chromosome 21. But our control group, the UK Biobank group, was not an ideal group. As I previously mentioned, the best control for this group would be mild COVID. These are people who have definitely had COVID or SARS-CoV-2, but did not develop severe symptoms. And at the time, we were not able to pick these out of UK Biobank. Therefore, we had to rely on a subset of people who had tested negative. For this reason, and for others, we replicated our results using other datasets. So we performed a meta-analysis with other genome-wide association studies to look for replication of the points in the genome that we saw associated with severe COVID-19. We looked at 2 different datasets, HGI, or the Human Genome Initiative, which had just under 2,500 cases and nearly 500,000 controls, and the 23andMe dataset from Google, which had just over 1,000 cases and over 500,000 controls. In both situations, these case groups were not as well defined as our genomic group. So we would expect some variation in the results. So anything that replicates within all 3 studies would be so any point that replicates within all 3 studies would have very good evidence for being associated with severe COVID-19. So here we can see our results from Genomic on top, and on the bottom we see the results from the meta-analysis. And we were able to replicate 1, 2, 3, 4, 5, 6 points within our meta-analysis. I'd like to make a note that the chromosome 3 locus, here shown by LZTFL1, was found previously in the Ellinghaus et al. New England Journal of Medicine study in the middle of last year, before we found it. And this study also identified the ABO locus on chromosome 9, which they found associated with severe COVID-19. We did not find this locus associated with COVID-19, and we didn't find that it was present in the other data sets that we looked in. So it's likely that this is associated with COVID-19 in general, but is not associated with that very severe requiring hospitalization form of COVID-19. Additionally, the point on chromosome 6, marked here as CCH-CR1, is within something called the HLA region. The HLA region has a lot of duplications and is very difficult to sequence. And for this reason, we didn't feel that there was enough evidence for this region to be associated with COVID-19 in our study. This left us with 5 points on the genome associated with severe COVID-19 to different degrees. So you can see here the significance p-values comparing our dataset GCC or GenomiCC with the UK Biobank UKB. And each point has a differing significance and therefore a different association with the disease. So now that we have points on the genome associated with COVID-19, that is, we have small DNA changes that we can be fairly confident are related to someone's risk of progressing to a severe illness, we wanted to know what genes are associated with the disease. And remember, this was at a time last year before very much was known about COVID-19, and people were still looking for treatments and trying to determine the best way to treat people who were severely ill. So we wanted to find the genes associated with severe illness, and to do this, we do something called identifying a causal gene. We identified the most significant single nucleotide polymorphism in a region, and here you can see it as this blue diamond. There are many other significant SNPs in this region, but this is the most significant. And although this is not a perfect way to identify a causal gene, because there is no guarantee that a significant SNP is going to be close to the gene that it acts on, there was a paper that came out in April of 2021, which showed that the closest gene is very often associated with the most significant SNP, and therefore is the causal gene. Using this method, we identified the IFNAR2 locus on chromosome 21. IFNAR2 is a receptor for type 1 interferons. It signals transcription of interferon-stimulated genes by reacting to type 1 interferon, which binds to it and propagates the interferon signal through the JAK-STAT pathway, leading to initiation of transcription in the nucleus. And interferon-stimulated genes are also known as ISGs. ISGs are the first line of chemical defense against viruses. So we can see how IFNAR2 may be associated with inflammation and reaction to the virus in a human. Secondly, we see the DPP9 locus on chromosome 19. DPP9 inhibits inflammasome activation. The inflammasome is a protein complex which causes inflammation in cells, and DPP9 therefore acts to prevent its activation, preventing inflammation normally. So we can see a situation where DPP9 expression might be disrupted, meaning that it can't fulfill its function of inhibiting inflammation, causing inflammation to become more apparent. DPP9 has also been associated with pulmonary fibrosis in other GWAS studies, and pulmonary fibrosis is a very common complication of COVID-19. We also have the OAS locus on chromosome 12 associated with one of our hits. The OAS locus contains 3 OAS genes, 1, 2, and 3. And the OAS genes are stimulated by interferon. They are one of these ISGs. They detect viral RNA when it's released in the cell. And when they detect it, they set off a cascade to induce degradation of viral RNA. So we can see a situation where disruption of OAS expression might lead to the virus being more able to replicate in the cells. So the story so far, looking at these 3 genetic loci, gives us 2 different sides to the same coin. We see the interferon response through IFNAR2 and OAS, and we see viral detection through DPP9 and OAS. But the story isn't always so simple as looking at the closest associated gene. There is something called linkage disequilibrium, which is non-random association of alleles. This means that nucleotide changes will be inherited together. On chromosome 3, at the most significant SNP, we can see that there are other SNPs that are also significant. Additionally, nearby, we see that there are a number of SNPs that are also significant. And when we do the statistical analysis, we can tell, using something called the linkage disequilibrium measure, or the R-squared, that these are very much associated with each other and are likely to be inherited together. So if a person has this significant SNP, they are also very likely to have these other SNPs. Therefore, it's difficult to tell which DNA change or SNP is responsible for the person being more likely to have severe COVID-19. At this locus, the LZTFL1 locus on chromosome 3, which we have just named for its closest gene for shorthand, we can see that there are many genes in this region. This is our most significant locus, with the significance being up to a negative log 10p of almost 30. The odds ratio is over 2, which means if you have this DNA change, you're over twice as likely to end up with severe COVID-19 than anyone else in the population. And we know that that changes in this region replicate across COVID-19 phenotypes. So that means that people who don't have severe COVID-19 are also likely to have changes in this region. And any of these genes could be involved. And I've specifically looked at CCR9, CXCR6, CCR1, CCR3, and CCR2 here, because these genes are associated with the immune response and therefore are tightly related to inflammation. To investigate which of these genes might be related, we have to take a step back and remember that not all tissues express the same genes. So within a person, as you probably all well know, there are different cell types, and each cell type will have the same genome. However, each cell type looks different and behaves differently, and that's because they have a different transcriptome. The instructions made in each cell differs despite the genome being the same. And so we change our question a little bit, and we ask, is X related to Y in a specific tissue, so within a particular cell type, where X is the DNA change or SNP, and Y is the transcribed RNA, or the instruction being made. We looked in particular at lung tissue or lung cells, because as we know, SARS-CoV-2 affects the lungs quite dramatically. We performed a transcription-wide association study, and you can see here that on chromosome 3, which is the region that we're most interested in with respect to this analysis, we can see that LZTFL1 is very low and has a low significance, whereas CCR3 and CCR2, along with CXCR6, are very high. So at this region, we think that these three genes are likely to be associated with severe COVID-19. We also see OAS3 on chromosome 12, which means of the three OAS genes, it's most likely to be OAS3 that's active in the lungs, or associated with the disease in the lungs. When we look at the direction of effect, so when the gene goes up or when it goes down, which one is more likely to affect the outcome, we see that CCR2 acts in the opposite direction to CCR3 and CCR6. So we think that more CCR2 is associated with severe COVID-19 and less CCR3 and CXCR6 is associated with severe COVID-19. So even though the SNPs are in the same region and they might be affecting cells in the same tissue, they are actually affecting them in different directions. So the genes are affected in different ways. To look into this more closely, we asked a further question. We asked, if I change Y, instruction of a specific gene, do you affect the probability of developing severe COVID-19? And we found that in the case of two genes, this was the case. The first was IFNAR2, which we've previously discussed, and the second was TIC2, and this was a gene that I haven't mentioned yet. The TIC2 locus was on chromosome 19, and we see that we have a significant SNP in this region. But we can also see that there are many genes in this region, and picking out which gene is likely to be responsible is difficult by just naming the closest gene. Luckily, Mendelian randomization has shown us that it's likely to be TIC2 that's associated with severe COVID-19. TIC2 is associated with this type 1 interferon signal. So when type 1 binds to IFNAR2 and IFNAR1 and activates the JAK-STAT pathway, TIC2 is one of the proteins that delivers the message from the cell periphery to the STAT proteins. The action of TIC2 can be inhibited by a chemical called baricitinib. And baricitinib is a broad JAK-STAT inhibitor. It's already used to treat inflammatory disease, so we know that it's safe for humans and we know what doses to give it in. And it's now in trials in UK hospital for COVID-19 as part of the recovery trial, which is a large randomized clinical trial of various therapies for COVID-19 treatment. So in summary, we've implicated the interferon response, we've implicated genes associated with viral detection. At the chromosome 3 locus, we think that it's CCR2, CCR3, and CXCR6 that are responsible for severe COVID-19 or associated with severe COVID-19. And we've also seen type 2, again, part of the interferon response. And we've determined that this can be inhibited by baricitinib as a potential treatment for COVID-19. As of this morning, we've now recruited over 13,500 people in the UK to GenomiCC. The results I've shown you now were based on the first roughly 2,500. So we are continuing to sequence these individuals and adding all the data together so that we can have a better, more comprehensive view of which DNA changes are associated with COVID-19. And over the next few months, we expect to get more information and be able to have more confidence in the results and the genes. I'd like to thank everyone that's involved in GenomiCC, which includes the administrative staff, in particular Fiona Griffiths, my co-authors, Erola, Lucia, Andrew, and Conrad, and everyone associated with writing the paper and analysing the data. I'd like to thank the Edinburgh Clinical Research Facility as they did all of our DNA extractions. I'd like to thank research teams at over 200 sites throughout the UK. And in particular, I'd like to thank my PI, Kenny Bailley, who's chief investigator on this project and on several others. Thank you for your attention. If you want to look at the paper for the GWAS, you can follow this QR code. And if you'd like to ask any questions privately or submit any feedback, you can do so at this QR code. And I'll take any questions if there are any.
Thank you, ma'am, for such a wonderful presentation. Our audience might have a few questions for you. So can we please have the questions? So the first question is from Naita San Faruqi.
I'd love to follow your research work. Is there any way I could do so? So you can look at the Bailley Lab at bailleylab.net, and we put up all of our publications on there. If you want to follow me in particular, I'm available on Twitter at dr_Sarah_C, so Dr. Sarah C. And I usually post updates there.
Okay, next one is from Radia Tumardia.
Does the coronavirus transmission and the severity of this disease rate depend on the continent due to variations in genomics? So we believe that that is the case, that there is variation in the genetic makeup of individuals from different parts of the globe. As you saw, we divided our input data set, or the participants, up into different ancestries, so European, South Asian, East Asian, and African. We also had American, which we left out because there were so few. But we do think that there is a difference in the genomes of people from different regions. And it's been seen in the UK that individuals from minority backgrounds are more likely to have severe COVID-19 compared to people from a European background, where a minority background is South Asian, East Asian, and African. So there is, whether or not this affects the transmission, I don't know. That's probably more to do with the viral genome itself, but definitely severity has been associated with the genetic background of the individual.
Okay, thank you. So,
what if we continuously wash our sinus by appropriate nasal sinus cleaner? Can we be able to get rid of the coronavirus? There has been studies that show that using sinus cleaners, especially those with saline, can be useful to remove the virus, because it gets rid of the virus out of the nasal passage. But I wouldn't rely on that as a method of avoiding the virus. You should still take good precautions like social distancing, washing hands, and wearing masks if possible.
Okay, do you have any other question? I guess we don't have any other question. So we're yeah. So we're at the end of our speech. Thank you so much. It was such a wonderful time, and it's really helpful for a lot of us. Thank you so much for being here with us today.
Thanks for having me.
And for everyone, stay safe till then, stay safe and keep your family safe too. Thank you so much.
Thank you. Thanks, everyone.
Thank you.
Bye.
Bye bye.